Thanks. And thank you, Elena, for receiving me. I'm hiding this week here in Seville so that I can finish this paper, or try to finish this paper. And Elena is hiding me from everyone in Berlin, so this is very nice. Thanks a lot. Yeah, so this question here, can RAD-Seq help sea turtles conservation? I want to start telling you that I'm not doing conservation. Yeah? So the idea is just that we come up with maybe some tools that can help other people do conservation. So I like to say I work on conservation genomics, but at the end of the day, I work on genomics. And my tools can be used if someone dares to use them for conservation. So the project and the data that I'm presenting here, this was all data from 2 different projects that were run by a former postdoc of mine, Sibeli Vilasa, who is now in Canada. And we had mostly 2 projects, one in phylogeography and the other in hybridization in sea turtles. The sea turtles, there are 7 living species, and all of them are endangered in different levels. And conservation genetics techniques already exist and are used for conservation or helping conservation of sea turtles in different ways. So for example, for the identification of the mixed stock, you know, the populations of the sea turtles, they are actually formed by the nesting colonies. But throughout the year, they go to the feeding areas, and there are the different nesting colonies they can mix. They can mix, and then in order to analyze where these populations come from, you do this mixed stock analysis. Obviously, population structure is also very important because you want to know if the populations, they are diverging, if they are exchanging migrants, and so on. Another fact that happens in sea turtle, which is very common, is hybridization between species. Usually this hybridization happens in a very low level, but there is at least one area in Brazil, on the shore of Brazil, where hybridization levels have reached over 40%. So over 40% of the females are hybrids between 2 species that are about 30 million years apart. And demographic changes over time, they are also important, especially if we are talking about recent demographic changes, if the genetic tools can help us to estimate them. The problem here is that most of it is done with single mitochondrial DNA genes or parts of the genes, or a limited number of microsatellites. There will be no problem with using these markers. The problem is that there is a lack of resolution. You need more resolution to define. The sea turtles are very similar to each other, the different populations, so the resolution that we have nowadays is not enough to actually define different populations and if there is structuring or not, and so on. So here is the phylogeny of the sea turtles. As I said, there are 7 living species, and they go all the way down to about 100 million years ago. Yeah, the most basal lineage, the leatherback, was living with the dinosaurs. At least when you see some pictures of dinosaurs, you can see a leatherback swimming there. This is pretty nice. And it hasn't changed much over time. Out of the 7 species, we have data for 5 of them. And apart from being representative of the phylogeny, these are the 5 species that have a worldwide distribution. Okay, so they swim in all seas and all shores. So now if we look at the phylogeny of only these 5 species, you will see that 3 of them are more closely related, even though this here is about the less common ancestor is about 30 million years ago. Then you have the green turtle, and you have the basal lineage, the leatherback. We have 1 draft genome for the sea turtles from the green turtle. And I want to really emphasize here that this is a draft genome. And the problem with draft genomes is that you cannot necessarily trust the quality and everything that is in their sequences. So about the sea turtles' molecular evolution. The history of sea turtles seems to have gone through a deceleration of evolutionary rates. So they have not been evolving in their sequences as quickly as other animals. So they all have the same number of chromosomes, and they are expected to have high levels of homology, which is just the content of the genome should be very similar. Not only this, but the content of this genome should be in the same order, mostly, and also the level of divergence between different species is expected to be very low. So that's why, even if you think about 100 million years ago, in terms of molecular evolution, it doesn't reflect that amount of time, that time of divergence. So here we have the draft genome from the green turtle. What? Okay, so here we have the draft genome from the green turtle, but there is another turtle which is unrelated, a freshwater one, that has a genome which is in a bit better shape. So as you see here, this assembly level of this genome is at the scaffold level, which means it's not assigned yet to the chromosomes. While here, it's not completely assigned to chromosomes, but it's starting to get there. So we could potentially trust this genome better than this one. So this is our backup reference for all of our analysis. So then as a summary here, we have the 5 species. We have a draft genome from one of them, and we have another reference genome that we can potentially use. So the methodological goal, given that we had data, population data from 5 different species, we wanted to use a high-throughput methodology based on genomics that could be broadly used for all sea turtles. Yeah? We were working with the assumption that they would not be so different because they have this genome structure that is so conserved and so, thank you. Not only in their content, but also in their structure. So we were using this as an attempt to have the, it works sometimes and sometimes it doesn't, as an attempt to use the same methodology. With this high-throughput methodology, we wanted to generate genetic markers that could be used for different evolutionary and conservation genetic related questions. So phylogeny, phylogeography, genotyping, species identification, but also paternity tests, and so on. So the methodology that we have chosen is one of the RAD sequencing methods. It's called the double digest RAD. This double digest RAD, I'm going to explain very briefly. It uses always 2 enzymes. That's why it's double digest. These 2 restriction enzymes, they usually have one short restriction site and one longer restriction site. And the short one is called the frequent cutter, and the long restriction site one is called the rare cutter. So the frequent cutter is going to cut the genome much more often. So it sort of controls how the size of the fragments after the digestion. And the rare one is controlling the fragments or defining the fragments that are going to get sequenced. That's because they only get sequenced if they have one restriction site on one side and the other on the other side. Because as you put the adapters for Illumina technology, you need to have 2 different adapters so that it gets sequenced. And in this way, you control how many loci you're sequencing from your genome. So you end up with a sub-representation of your genome, even though you can sequence sometimes about just 1% of the genome, because the restriction sites, they are supposed to be throughout the whole, all the chromosomes, you are subsampling throughout the whole genome. So you can have a good idea, not only over one region, but what is happening in the variation throughout the genome. Does it work? Yes. For the double digest RAD or the DD-RAD, you can also use size selection. So you establish a size range for these fragments so that you have another control on the number of loci that you're going to analyze. By loci, so one locus is just one predefined position in the genome is one locus. So loci are more than one locus. So here, for example, if we chose the size range, we might have 3 of these loci that we are going to sequence. But in some cases, I don't know if you can see, if it happens that your restriction site has a mutation for one of your alleles, so this is T, this is a allele with a T in this position, and this is another allele with a C in that position. If this one has gone, has also a polymorphism or a mutation in this position, what happens is that this fragment is going to be this size and not this size. And then you're not going to sequence it if it's out of the range that you have pre-established. So you're missing this allele. In order to control that and to know, so what should I use? What enzymes should I use? How should I choose the size range and so on? If you have a genome, you can use this genome to sort of simulate your experiment in silico. So we performed an in silico digestion of the green turtle genome with the two enzymes that, well, we performed with many enzymes, but this is the results with the two enzymes that we used at the end. And that's how it looks like. So we had the whole genome, all the chromosomes, and after digestion, most of the fragments are quite small. So this is 50 base pairs, 100 base pairs, and so on. So you can see that as you go longer the fragments, the fewer loci you were covering. Yes, they directly, so this here is directly represented by one specific fragment length. So if you choose a range, you are basically predefining how many loci you are analyzing from your genome. If you choose another range, you are choosing a completely different set of positions of your genome and a different number of loci in the genome. And this is exactly the range that we have chosen because when we were doing the in silico analysis, we were trying to find a range where we could get between 30,000 and 40,000 loci from the genome. So we knew it wouldn't be exactly that. And at the end of the day, we could select between 500 and 600, but this includes the adapters that we are adding. So when if this is what we are selecting in the lab, you have to discount the adapter sizes so that you actually have the proper sizes from the fragments from your genome. No, this works. Okay. So, and what have we done? We chose, I think you saw here, we chose pretty long fragments, 400, 500 400 to 450 more or less. And usually in Illumina, if you go to the biggest machines, what you sequence is about 100 base pairs. So most of the RAD experiments are done with just 100 base pairs from one of the sites. So you would be sequencing maybe this first bit here. Yeah? Sometimes you sequence the first bit and the last bit. So this part and this part. So you don't know, if you don't have a reference, you don't know how they relate. You don't know how far apart they are. So you actually don't know the size of your original fragment. And we wanted to know, we wanted to have this control. So we chose a technology that would give us very long reads that we could overlap and recreate the entire fragment from the genome. So at the end of the day, we had long haplotypes, average 400 base pairs, instead of just getting SNPs, which is what most of the people try with RAD. They try to get one variable position so that they can do several tests afterwards. This one. So why did we choose long haplotypes? The long haplotypes, so first of all, the sea turtles, they have high levels of shared SNPs. As I told you, they evolve a bit slow. They have some levels of hybridization, so they can share some SNPs because just over time, over the evolutionary history, they have been exchanging. Or maybe they even didn't have the time to fixate all SNPs to one direction or the other direction. So when you work with long haplotypes instead of one single position, you have a better chance of actually assigning these to species A or species B, or population A, population B. The haplotypes, they give more information than SNPs. In a haplotype, you have the actual sequence. You have all the invariable positions, not only the polymorphic positions. You can do a lot of analysis that would not just mean that you are getting all the variable positions of the genome and throwing them, but you know actually you can calculate mutation rates, substitution rates, and so on. Several population genetics software also still take haplotype alignments instead of SNPs. So if you want to do certain types of analysis, you cannot use SNPs for those, but you cannot always take the SNPs out of the haplotypes, but not the haplotypes out of the SNPs. And they can be used to develop primers. So for conservation, we cannot expect that people are going to have access to next generation sequencing machines. They might want to use primers, they might want to use microsatellites, and for these, they are going to have long sequences that they can use to develop primers for their specific goals. Also, one of the biggest problems when analyzing RAD is because since you don't have the whole sequence of the genome, you are picking just these very many positions of the genome. You don't know how many of these positions represent paralogs. Paralogs come from duplications in the genome. These duplications can happen in small bits of the genome, entire chromosomes. It depends. Each species is going to have its own history. And you don't know beforehand how much duplication happened when and how much polymorphism or variation they have been accumulating over time. But the problem is that paralogs, they represent different positions in the genome, but when you get the sequences, they look very similar. So you might end up putting them together and analyzing them together, even though they are not homologous. They come from two different positions. How to get these long haplotypes? We use the MySeq, so a specific machine from Illumina, and we use the paired end 300 base pair, so the longest reads that exist. We did, or we tried to do a precise selection using a machine called blue pipping, which is then run in a, well, sort of pulse field thing. I don't know how to explain that, but it's supposed to be precise. And also our choice of enzymes was driven by our in silico digestion. So here I have again the phylogeny for my 5 species. And just, okay. I think you remember, so I'm using the same approach. And you remember that it's thought that they have high levels of homology, that they have very similar content in their genome. And this is something that we can test. It's the first thing that we decided to test with the data that we got. So what we did, we have, the data. Okay, the data for each of the 5 species, and we had a draft genome. So we did a blast, which just means we are comparing by identity our sequences to the draft genome, and we were looking how many times we would have a hit. So some sequence that was somehow similar to the sequence in the genome. When we didn't have a hit, then we would use the more complete genome just in case something was missing in the draft genome. And these are the results. For the 5 species, so this green turtle is this here, okay? We got basically very similar content in terms of comparison between what we got and the genome, about or around 98% for all of them. So almost all our sequences have a homologous in the genome from one of these species. So yes, they have very similar content. Just now, I have to say something that the DG-RAD, or when you use RAD, because you're using these restriction sites, you are biasing the sequences of the genome that you are analyzing towards high complexity regions. The high complexity regions is not that they're very difficult or complex, it's just that they have a good mixture of nucleotides. You have regions of low complexity. These are the regions that have, for example, high C content, high GC content, a microsatellite that is very, very long, repetitive regions mostly. They are very low complex, and we are selecting against them when we use 2 different restriction sites, okay? So we are basically analyzing the high complexity part of the genomes, and these we can say the content is very stable for sea turtles. We don't know about the repetitive part based on this data. And then the bit that was missing here, we compared. Can you still see with the lights? Yeah. We compared with the freshwater, more complete genome, and then we got a bit more, yeah? Just a tiny bit more. We still had a few sequences that didn't match any turtle, and so we compare them to the public database, everything that is in the database, just to see, do we have some contamination there? What is there that is just 1% that we cannot catch in the genome? So we still got some turtle sequences, it's just that they were a bit too far apart for the thresholds that we used before for the identity, but we got some bacteria as well. Here, this is all bacteria, which is fine. It's something, I mean, we had a skin sample, so we expect to have some bacteria in there, and we did find. So the one that we find a bit more bacteria than the others was the one that had actually more than 1% of sequences not in the turtle genomes. And this was because it had a bit more contamination, not because it has a lower content, turtle content. Okay, so this was the first bit. We knew we didn't have contamination or very little, so we didn't have to worry about this. And then we just ran one of the RAD pipelines. There are many pipelines nowadays. At that point, when we ran the first version of this one called PyRAD, and we got results. Because when you throw sequences in a software, you're going to get results. Just remember that. It doesn't mean the results are true and that the results are good, but you're going to have results. So with these results, we could see several polymorphic microsatellites, which could be really interesting for analyzing, for designing primers. We could identify hybrids of first generation and even back crosses between different species. We found thousands of SNPs that were diagnostic for the different species. So these ones separate that 3 species that are closely related from the other 2. And then another bunch of SNPs separate these 3 here. This was not a problem at all. So comparing among species was really easy with this data. We also could calculate what was the intraspecific nucleotide diversity. So was there one species that had low variability compared to the others? And yes, so we found that the green turtle was the one with the highest level of nucleotide diversity, while the leatherback, so the basal one, was the one with the lowest. So there was very little variability in this one compared to the others. But when we got to doing population structure analysis, our data was no good. Like, we couldn't find one single population structure. We know sea turtles have low levels of variability. So we knew we needed a lot of data, but we need good data. The data has to be reliable. If we have artifacts of all kinds, we are not going to have results there. And so we didn't get. So we decided to go back and try to control every single step so that we would improve our data and remove all the artifacts, or most of them. So when you do RAD analysis, you have 2 possibilities. One is that you do a de novo, so you don't use any genome as a reference. And here you just get, the first step, you get your reads, and you cluster them by identity. So you define a threshold, identity threshold, and you cluster them by identity. And then you assume at first that, oh, this represents one locus in my genome. This is another locus, and this is another locus. And then you try to genotype to see, is there 1 allele or 2 alleles? They are diploid, so the maximum is 2 alleles. That's basically what you try to do. If you find 3 alleles, you're going to get suspicious. It's either a paralog or you have some errors. And the other is reference-based, which is much easier. You have a genome, you map everything with a predefined threshold, and then you just call your genotype. You call SNPs in all these positions. But for that, you have to trust the genome. The essential requirement here is that you have a collection of reference single copy loci. So if you have loci that are repeated in the genome, your reads are going to go sort of randomly to one or the other. You're going to have a lot of ambiguous signal there. So you want to, your first goal is, I need a reference or I need a collection of loci that are single copy, just present once. And yes, the problem here is that it's difficult, and there are many, many errors that you find on the way when you look very closely to your data. And I'm from Brazil, and in Brazil we say cockroaches, if you see one, What does it mean? Arrows are like cockroaches. You see one, you might want to kill it, but if you see one, it's because there is 1,000 more. So you don't kill the arrow that you see, you kill the source. You have to go back, look for the source, and kill the source, because you might be sure there are tons of arrows just like the one you found by chance. And that's what we try to do. So I'm going to tell you some of the cockroaches that I found, not all, because if you want to know all, if you want to use this technique, we can talk later. So the first thing, we need to find the threshold. So I told you about identity thresholds that are important to define that sequences belong to the same locus. And this is like the first very important decision you have to make. There are many ways to find these thresholds, and usually you just pick one, you test it, you pick another one, you test it, and then you see the final results. Yeah? It's not exactly the best approach, but it's the best that people could do up until now. So we have this region of the DD-RAD. We had a blast, so, okay. To identify the threshold, we thought, oh, we are going to do an in silico, no, we are going to get the in silico results, and we're going to compare to the genome and see, so what are my paralogs? How do they look like? So does it just means that, I think here I have something. If you compare the sequences that you got from your own genome back to the genome, the best hits that you get are the same fragments, because you digested the genome and you are using it again. But the second hits that you get, so the second more similar hits, they're going to be the second locus in your genome, or the second position in the genome that's the closest to yours. So in many cases, you're not going to find anything, because one certain position is very different from the rest. But in many cases, you're going to find. And what we found was something very weird. This is level of identity, so from 80 to 100. Here, I don't trust below that because of the thresholds that we have used. But from 80 to 100, I would expect some distribution, like duplications happen over time, insertions, transposons jumping around the genome. So you're going to have some level of duplication, some level of paralogy. But I didn't expect this. This was like, for, I don't know, at least 700 of my loci, I had an exact same copy in my genome. So as if my genome have had some large duplications. And this was really weird. So we decided to then check the genome a little bit deeper. We got another draft genome from another turtle that was published together with this one, and it had the same pattern. And this was funny. Like, was there some duplication in the turtle's genome that nobody knows about? And then we got the more complete genome, and the pattern was completely different. And by investigating this, we actually realized that when you have draft genomes, many times the way you build them, so you assemble them, can generate artifacts, because you have lower coverage, because you have very uneven coverage throughout the genome. And these artifacts, sometimes they make copies of the same position again. And this happens again and again. It could not be a problem many times, but if you don't even know it exists, it can become a big problem. So if I have 2 copies of my reads, what's going to happen to my reads? Are they going to go to one, to the other? They're going to split? That depends on how you do your analysis. So you might lose everything, or you might do comparisons that are going to have 2 completely different lines, even though they should have one. So we decided the draft genome not to be trusted, not yet. So we need to understand more so that we can use this reference. So we decided to do everything de novo, so with our data. For that, we had to choose a threshold. Yeah, as I said, to build these different loci or to define the different loci, we need to assemble or cluster the sequences by identity. So for that, we got our best data. So the high coverage DD-RAD for all the 5 species. We have 2 individuals of each species, so we had 10 in total. And we had, for each of the individuals, they were in 2 different runs. So they were done independently, with one exception. And they came either from one project or the other, the phylogeography or the hybridization. Here I have then 4 different runs. This is just an example. This is not all the individuals in the runs. But just for you to understand, this is like the shape of the fragment length after we have merged the reads. So remember, the digestion of the genome was like this. But I wanted to size select a certain range. Yeah, and now I'm just seeing, well, what did I get? I know what I wanted to select, but one thing is what I wanted, and the other thing is what I actually did and what I got at the end. So this is the size selection that was attempted for each of the runs. You can see you have a bunch of small fragments, sometimes a lot of them, that escape somehow. Sometimes you have things that are a bit out, so they're off what you wanted, right in the middle, from one side, from the other. That's basically your loci dancing. You're getting different collection of loci for every single run that you do and every single library, because within each run, we had sometimes different, we pooled the individuals in different libraries, and then they behave differently. So this is something that people usually don't know, because they just sequence one side of the fragment, they don't know the actual size of the original fragment that they had. So they never have this shape. And basically, they get all of this and they throw together in some analysis. And they don't know how many loci they are actually focusing on the genome. They are targeting in the genome beforehand. With this, what we did, we defined what was the range that was very well covered in all the individuals, and we did an in silico size selection. So we had the data, then we just cut out what was out of our range. So at least we had some sort of control. This is one example of one individual and 2 different runs. Yeah, so this is what I got in one. Here I actually selected twice the same library. I ran it twice through size selection, so it was very narrow, more than what I wanted, but at least I didn't get these guys here, like I got in this case. And when we merged for each individual, so 10 individuals, all the data that we got, this was the shape. Yeah? So I had peaks all over the place. I had a bunch of small fragments in different levels. So this is what the region that we have decided to use. So we did the in silico size selection. But still, if you see, I have here very high peaks and sometimes very low peaks. And now is the problem. If you have a lot of data from one individual and little data for the other, you're going to generate artifacts. Because you're trying to treat all of them the same way. You're finding the same thresholds. You're using the same pipeline, the same parameters for things that have a very different amount of data. And a different amount of data means different number of errors. The more you sequence, the more sequence you're going to have that have errors. Even though proportionally they should be similar in terms of absolute numbers, they are very different. So what we have done, maybe I have to say this now. We normalized. We tried to get the same amount of reads for each of our individuals so that we could treat them the exact same way. So what were the thresholds that we had to choose here? The minimum coverage per allele. The minimum coverage per allele separates real alleles from sequencing errors. The errors, you assume they are very rare. Like usually they're going to be in one sequence, maximum 2. Obviously, it depends on the total amount of sequence that you have. But statistically, for what we were aiming, 1 or 2 were mostly errors. Minimum coverage per locus, which will define if you can trust or not your genotyping. So if you have low coverage, you cannot really trust that you got the second allele. If you have high coverage, you have a better confidence about this. And the maximum coverage per locus. Because when you have data that is repetitive and they all cluster together, the amount of sequence is going to accumulate, right? So you end up with a lot of sequence for one locus, which usually means that it comes from different positions in the genome. So yeah, so here is just showing that we have normalized for that. Because if I would say my minimum coverage per allele here is 2, and here is also 2, I have so much coverage here that I'm sure that some errors are going to get frequency more than 2, just because I have too high coverage. So our results here, when we called maybe I should just show this quickly to remind you. Oops. OK, here. So we were clustering our reads, and we were forming the different loci, yeah, that we assumed were the loci. We did filtering and so on, and at the end, we had a number of loci. Remember, at first, we were aiming at 30 to 40,000, but as we did, our size range was a bit smaller. The in silico one, at the end, we were aiming at a bit less than that, about 20,000. And that's what we got. So here I have the 2 individuals from the green turtle. I have the genome from the green turtle, like I did an in silico digestion, so I did the simulation. And here is the same data, but that has gone through my pipeline. So the pipeline to remove paralogs and errors and so on. And when you look at the, and here's fragment size. So the longer the fragment, the fewer loci you have, about 400. The shorter the fragment, the more loci you have. So the shape looks really nice. Like my data and the genome, they look pretty similar. So I was happy about that. But it doesn't mean I have exactly the same loci, but just the same number of loci. When I look at all the 10 individuals, mostly the shape is also very similar. I have one very strong a different one here, which is one of the guys, or the only guy that had very low coverage for this region. I couldn't reach a better coverage for this one, and this reflects in the number of loci that I caught for this. But the others, where we tried to reach some sort of saturation, meaning we tried to catch all the loci that were there, they were following pretty much the same thing as the genome. And if the turtles are so conserved for this high complexity part, that's exactly what I expect. The numbers should be similar. I have one exception here, because this is 2 individuals from the same species, but exactly at this size, there was a peak. It's some repetitive something that was exactly this size, and because I gave the exact same number of reads, the total number of loci dropped, but I corrected for that afterwards. And then, so after we compared the number of loci, what about the content? Are they the same? How much do they overlap? And here, so here we see the phylogenetic tree again, just to remind you, because this reflects pretty much the results. Here on top, I have the total number of loci. This is 20,000, okay? So for these are 9 individuals. I excluded the one that had lower coverage. So 9 individuals. For basically all of them got between 22 to 23,000. They're the top ones. The second one is always the other individual from the same species. So this is the same, just with the opposite. So this is the total number, and this is how many they have in common. Total number, how many they have in common. Okay, so you can see that sometimes it's always very similar, but sometimes it's more similar, like these ones. Sometimes it's less similar. And this completely is reflected by the level of variability within the species. So these guys that are very similar, this is the leatherback, low polymorphism, and this guy that are more different, this is the green turtle, more polymorphic. So this reflects, because the more polymorphic the species is, the more mutations you're going to have in the restriction sites. Yeah? So the overlapping between loci drops. So the very polymorphic SNPs, I would not use this approach for very polymorphic SNPs, no, species. I would not use this approach. You have to be very careful. And then the rest really coincides with the tree. So here I have all these 3 species. They always cluster together around 17,000, 18,000 of loci that overlap, that are homologous. Then I have this guy against all these 3. And I finally have the leatherback here against all the others. Yeah, so for all of them, I had about 7,000 to 8,000 loci that using the same approach that are now homologous and comparable. So it's still something nice. 5 different species, 100 million years apart. I have 8,000 positions in the genomes that I can compare, and that I can be more or less confident they are not repetitive. So with the results that we got so far, we can generate diagnostic markers for each species. We can define already variable microsatellites intra-specifically and design primers. We could determine the genetic variability among the different sea turtle species. And we could build, that's the most important result in my point of view, very reliable references to be used for population analysis using the RAD. So remember my first result, not good for population structure. Now I got to the point where I have a nice reference and I'm going to try again. Unfortunately, I cannot tell the result because I haven't done the analysis yet, but we are almost there. So the next is going to be the population analysis structure, hybridization and introgression. So we can easily identify first-generation hybrids, back crosses, but we want to go deeper and look at introgression over the historical evolution of the sea turtles. And the new reliable references. So I'm also applying DD-RAD for other species, so manatees in Brazil, and some monkeys, so woolly monkeys, tamarine, and sloths. I have many projects with the sloths. So mostly of them are then in Latin America. And one thing that I'm starting is doing meta-barcoding. I have several projects that I collaborate in meta-barcoding. And the idea for the meta-barcoding, that's when you get an environmental sample, and then you have a DNA, you PCR, and you have these barcodes. Yeah? You have a collection of barcodes. That's why it's called meta-barcodes, because you try to get all the variability of barcodes that you have in one single sample, and you try to assign to different species. So we are doing this using environmental DNA, and also iDNA, which means invertebrate source DNA. So when you catch insects that might be carrying DNA from hosts of some kind, in our case, mammals or vertebrates in general. So the blood-fed ones, or the ones that go on carcasses, the ones that go on feces. So we are trying to detect the biodiversity of vertebrates in a certain area through the insects. This I just wanted to mention because I am starting with several projects related to this. Yeah, and the acknowledgements, especially to Sibeli, the postdoc, Max, who's my bioinformatics student assistant, who is helping a lot, and all our collaborators who gave us samples and also money to do the research. Yeah, thank you.